Thank you very much for coming to the Cancer Center seminar today. And today's speaker will be Dr. Justin Hwang from Department of Medicine, Division of HOT. And let me, give some brief introduction. So it will be really brief because actually I want his data to tell about him. So then he's a graduate of Carnegie Mellon in Pittsburgh with BS and did a PhD in Tufts for biochemistry. After that, he joined the Dana-Farber, then did another postdoc at the MIT Broad. And after that, 2020, he joined U of M. And it is really privileged to have him here. And now he currently have involvement in many different aspects of clinical and basic research in our university and cancer center. We're represented by his NCI R37 Merit Award. So then I just want to, transfer the podium to Justin. And now podium is yours.
All right, everybody, can you hear me? All right, I put a tacky title for the last talk of this year. Examine our patients through magic. There is a lot of magic involved here. However, it's mostly related to data, in case you're looking for real magic. However, I just want to welcome any sort of questions during this talk. Mostly, in all seriousness, is all related to our cancer patients and how we can expand the knowledge that we're learning here in this very institute, and really expanding that out to affect understandings on a national level. And if at the end of the talk you feel like we still haven't made an impact, please tell me as well. All right, here's my disclosures, and here's a narrative of what I'm going to discuss. First of all, what is MAGIC, Minnesota Annotated Genomics and Cancer? This is an effort. Originally, it was meant to be a data portal. It became a systematic effort that works of us working with industry, clinicians, and sort of health regulatory officials, and the Office of General Counsel, IRB, all together to put something together so us as cancer researchers can start examining our own patients' data. And I'm going to present some case studies, which I find to be very illuminating, that help us really understand not only what's going on in our patients here, but how we can affect a sort of global scheme of trying to understand, for example, disease that I care about. Today, I'm going to talk about prostate cancer. And subsequently, I'm going to take some really deep dives into transcriptomics, a very big interest in my lab. I'm going to talk about how we can integrate different methodologies of understanding machine learning and how to really pick out key elements using transcriptomic data, as we know, has not been utilized as much in the biomarker and predictive space as much. And finally, I'm going to give a key example of how we can utilize transcriptomics to predict patient outcomes. I even think about this going forward, even in the setting of liquid biopsies. All right, so many of you know this, some of you may not, so I apologize if you already heard some of my talks before. Where are we today in terms of patient diagnostics? Right now, for a lot of our patients, especially in certain areas, we're really collecting every single tissue sample when we fail to have a treatment for the patients. We're sending to, many of the physicians will definitely know this in the sense that we're starting to send this to, for example, whole exome, whole transcriptome sequencing, and also getting genetic data from the same patients. This is telling us, of course, what is happening, what is driving the disease for specific patients, how they maybe some reasons why they're not responding to certain therapies. So this is done currently at the University of Minnesota. It's done for a lot of our patients already. Some of the partners that we use include, for example, Tempus, Caris Life Sciences, and Foundation Medicine. I'm going to go through some of these as examples today. Now, I really want to emphasize just one element when we get to these sort of diagnostic tests. And I started off my postdoc doing, my best study at the time in 2017 was paired 7 patients pre-treatment, 7 patients post-treatment. At the time, I thought it was remarkable what we could already find. Today, for example, our collaboration with Caris, and thanks to our leadership here, we have partnered with Caris to actually access their data portal. As I said, we have access to data from almost 250,000 of patients, and each patient, there's 1 million data points. Let's keep that in mind. So back in the days, I had 7 patients, and I was already finding crazy things in those 7 patients. Now we reached the era where quite frankly speaking, it's very hard to understand what is coming together. And if anything, we do have to really think about sort of data-based approaches to understand what's going on in our study, in our patients. So here's some, just to give a pitch to our lab. And up front, I would like to thank many of the individuals that have been involved in our research program. Some of you guys are sitting here, thank you guys. That said, it really involves a whole team. Sometimes this involves clinical trialists. For example, on the top right picture, you see Dr. Ryan, Dr. Interakas, Dr. Zorko. All these individuals are helping us think about patients from the clinical side. We have to partner up with, for example, people at the industry. Caris Life Sciences here. This is our meeting with them in ASCO. And it requires a whole lab to do this because we really have to think about all these different questions, how they come together and impact our patients. So our lab really works on the downstream of what we receive at the Masonic Cancer Center, which we receive all the data, we house the data, we work with a sort of BPIC team, which is our sort of unbiased broker, to connect this with the clinical health records so we can actually really understand what is happening in our patients. Now, so here's some approaches that I'm going to talk about throughout this discussion. I'll get to this in much more detail. Our lab is highly interested in, for example, machine learning, taking big data points and really reducing it to simple outcomes. For example, who is a survivor, who will not be a survivor. I'll get to this a little later bit of the talk. We also have interest in really deconvoluting the gene systems, which are very dynamic as we're starting to see in patients. If we look at patient to patient, let me just give you guys a thought out there. If we look at a prostate tissue versus a breast tissue, I'm almost certain that a transcriptional program is very different between the two tissue types. Yet when we consider these, for example, hallmark signatures or whatsoever, we're treating these pathways almost identically. So our job, at least in part in our lab, is to really say, how can we capture these nuanced differences between cell type using data alone and driven by data alone? Now, many of you know prostate cancer, some of you don't. I'm going to distill it to the most simplistic single slide. This is not going to be accurately represent all the disease stages. Some people will be very upset at me afterwards, especially if you're a clinician. But listen, I'm going to just mention loosely, prostate cancer still is leading to significant amounts of death in 2024. We estimate about 34,000 patients will die from prostate cancer, mainly from metastatic disease. And, I see prostate cancer as 3 stages that are relevant for clinical treatment. For example, you can think about the localized disease stage. Back in the days, we see a lot of these plots or whatsoever. Really, there's a lot more available for each stage of these patients. This is not like, for example, 2012 anymore, where we only have a few therapies available. Still, when the patients progress, we reach a stage where they may be sensitive, for example, to hormone therapies. For example, we call this a more hormone-sensitive metastatic disease. However, I really will focus on a lot of the late stage, for example, MCRPC, which is when the patients have blown through all sorts of current therapies. Essentially, they have very limited therapeutic options at this stage. Very much likely, most of them will eventually die. So why should we study our own patients? There's clearly a disproportionate representation of cancer if we just look at geographical location. This is a slide provided us from the Cancer Center. So thank you for the Cancer Center for providing this. You can look at Minnesota up here. We have a lesser rate of developing cancer and mortalities. However, we have a higher rate of developing prostate cancer. It's hard. I'm having a hard time seeing my own slides here, so just forgive me if I'm pointing at the wrong thing. Obviously, there's higher proportion of breast cancers as well as a suite of, for example, melanomas and blood-based cancers. So altogether, we might wonder why. And I would argue one thing is a factor without doubt. For example, race, ethnicity, socioeconomic factors, dietary restrictions, and basically the type of people in our community will affect the rates of the development of cancer. So really, it is important for us to think about what is going on in our community. The question is, can we do this? And how have we been doing here? So I'm just going to iterate one test. I will talk about other tests throughout this particular presentation. Really, throughout 2021 to 2022, 2023, we have started to collect massive amounts of Caris tests. You see there's strong diversity. Of course, there's driven by certain people in the clinic. For example, some of my favorite colleagues are collecting Caris tests for prostate cancer patients. So therefore, for example, you see 70% of these, 17% of these tests are all based on Caris tests. And what can we do with this data? We can start thinking about what is going on in our clinics once these patients fail therapy. Of course, there might be some other cancers here that you might have interest in, and we're happy to talk about this offline. However, what is the problem here? Now, I'm just going to point out, for example, all these tests. For example, if you receive a testing result, what comes in this report? Obviously, this report is given to clinicians. The clinicians probably don't have the ability to examine all the 20,000 genes of the genome and say, oh, okay, there's maybe one mutation in one of the 20,000 genes. That is simply not practical. So with that said, a lot of these tests, what they do report is a very simplistic version of what is going on in the patients. For example, in a foundation medicine test, they really focus on 324 genes. Decipher tests, interestingly enough, even though they look at 46,000 genes, they really only report the results related to 22 genes. With that said, you can look at other panels as a Genie and Keras, even though they sequence a whole exome, a whole transcriptome, they really have a sort of hyper panel that they look at about 592 genes. With that said, nobody's giving me sponsorship for this talk, so I'm not advocating for anybody. I'm just presenting some data that is available to us. With that said, as a data scientist, you'd be very disappointed because all you're seeing is, wait, why are we making decisions based on such a small fraction of genes? To me, it seems that there's a lot more to learn. I'm not going to make any more comments other than that. Now, this is an effort what we now call MAGIC, Minnesota Annotated Genomics and Cancer. This tagging name was not invented by me, but Dr. Antonarakis, who is online somewhere. That said, he indicated, so I'll just tell the story here. So there's one day, and I was fortunately invited to do a Keras site visit. Well, that said, when we did the site visit, they told us, Dr. Hwang, how come you guys are not taking back any of the data we've done for you for sequencing? And this involves almost thousands of patients. That time I was wondering, well, why aren't we? So with that said, I quickly talked to my colleague who was also at this visit, Emil Liu, who indicated that we had to find a systematic effort to bring back this data so we can study our patients. That said, eventually, obviously, Mano is in big support of this. We worked with Steve Johnson, who's our unbiased broker of the data, to really import this process. So I'm highlighting on the left the process. It might seem like a very simplistic workflow. However, in the color boxes, you can also see that these are different entities within our institute, outside the institute. And if you haven't run a lab or an institute, you understand that each one of these data transfers requires to have a secure shelter, a secure way to transfer the data, and sometimes RB agreements, and other times approval by the Office of General Counsel. So with that said, I am excited about this effort, but this has been a learning about how our institute works in sort of managing health records. That said, I'll quickly summarize this. Of course, from our patients, we get in our Masonic Cancer Center, we can get them, send them for tests in Keras, Decipher, Tempus, whichever you may want to sequence at. That said, they have already set up agreement. We have already been able to bring back the data into a secure server. With that said, now comes internal hurdles. With that said, once the data gets to BPIC, which is best practice and sort of, I think, integrated computational sciences run by Steve Johnson, what BPIC is allowed to do is pull Fairview data in a de-identified manner and then combine it with each one of the patients that we have genomic data sequence. That said, now that gets all sent into a BPIC data shelter. Unfortunately, this process is now approved by this type of test. For example, for Decipher, we have a Decipher data shelter. For Keras, we have a Keras data shelter, and on and on and on. You might ask, why are we making things so complicated? Because long story short, it really seems like the Office of General Counsel wants to approve and work with each one of the companies to indicate what is a feasible test for research use. So, and with that said, that has been a major hurdle, but we're starting to see some success. I promise you that throughout this talk, this will be the least magical part of the talk. Everything else will be so much better. With that said, finally, what can we do with this data? We can use it directly for research. We can play with this raw sequencing data to do, quite frankly speaking, I think, amazing things. That said, we're also, with a lot of partnership, constructing a local CBioPortal instance. And as you may know, if you have played with CBioPortal, this allows any person without informatics expertise to look at the data, to connect it to clinical health records, and also model and determine if some of the genes or sort of processes that you have been interested in actually affect our patients here again at the Masonic Cancer Center. So I'm just going to summarize the efforts here that we're doing. For example, we have constructed the CBioPortal instance. It is operational. We're hoping to hand the administrative effort to the Masonic Cancer Center, so my lab doesn't have to manage this, but I do think is a systematic effort that should go beyond my laboratory. we have started to acquire data thanks to Steve Johnson and his group. We have been able to pull in data into a secure server and shelter. All this data is essentially waiting and ready to go as soon as we get IRB approval for some of the research uses of this. That said, you can kind of see we already have a diversity of types of data. For example, Karis testing, Garden, Andrew Nelson has gene panel. I forgot some of the tests that we also looked at. For example, those of you interested in breast cancer, we also have been able to pull in the I-SPY2 data, which is also available to us as well. That says currently we're in the massive elongated process of working with IRB to address each one of their comments so that we can do research on these patients. We're modifying the data transfer process so we can protect us against state laws so that nobody's going to sue us for studying our patients. I'm sure we're not trying to identify them to steal their money, I assure you that. That said, lastly, we've already done some work on Decipher tests. I really think Decipher test is a very interesting test that is not approved by the NCCN, the guidelines that oncologists use in the primary prostate tumor setting. I'm gonna show some comparisons of what we see in the prostate cancer data. You guys can think about this. Decipher test now give you a risk score, starting from the first instance of disease in the urology clinic to tell us whether a patient will get metastatic disease up to 30% accuracy, not super high, and also thinking about whether they should be treated at this particular stage or not. So to that degree, this type of data is already being transformed into using for a clinical decision, albeit the use has been certainly limited. Now, I'm going to talk about a few patients in our clinic. Excuse me. All right, I'm going to talk about one of our patients. I'm sorry, my slides got disorganized a little bit. I'm going to talk about one patient that we found in our particular clinic. That said, this patient was brought to me by Dr. Chuck Ryan, who used to be the division chief of the Hematology Oncology Transplantation Department. He told me that, Justin, I found a patient with a BRAF fusion, TMPRSS2 fusion. And he said, is this weird? And I told him, well, I've never seen a TMPRSS2 BRAF fusion before, but as many prostate cancer researchers know, that TMPRSS2 fusions are seemingly selected for in prostate cancer patients. Almost all the TMPRSS2 fusion events are in prostate cancer patients. Interesting enough, we looked at the protein a little bit in terms of the fusion event. I'm just going to verbally summarize it. It seemed to be an event where, for example, the TMPRSS2, which is an AR-activated promoter, is linked to the kinase domain of BRAF, allowing hyperexpression of a BRAF protein. Interesting enough, this patient blew through a lot of therapies. I just want to point out this particular time point here, in which they received a MEK inhibitor, or BRAF inhibitor, and they somewhat responded. However, the patient did not tolerate this well at the time. And we had to stop the therapy. The patient eventually died. Coincidentally, at this time, another patient, somebody else in Brazil, published a case study indicating that they also observed a TMPRSS2 BRAF fusion, and the patient in that case survived after receiving some form of MEK inhibition. So altogether, we're thinking, wait, is this something that we should look at in more patients? I think the argument is, well, we should at least take a look. In this case, we partnered with two individuals that I had the fortune I still work with, of course, Emmanuel, and of course, Alexander Shirazi Raffel, who is from City of Hope. He's a faculty there that really thinks about clinical trials. He is very excited about BRAF, but he wanted to see more data. Long story short, we're finding that BRAF, this is a partnership with Foundation Medicine, which eventually looked at about 16,000 biopsies, tissue biopsies, and about 8,000, close to 8,000 liquid biopsies. However, there's something very unique about these patients in the sense that they don't exhibit your typical sort of class 1 V600E alteration. And I'm also glad I'm talking to a cancer audience, so you guys maybe know some of these BRAF mutations. That said, the V600E, there's targeted therapeutics, but however, in class 2, which we're seeing in orange here, and the other form which we have captured in our two patients, the TMPRSS2 BRAF fusion, are also the predominant forms of BRAF alterations found in both tissue biopsies, and we can detect it in liquid biopsy using the Foundation test. That means that in these 3%, approximately 3% of patients, we can capture if BRAF alterations are within the blood of cancer patients. Some really interesting things that we also found that when we looked at across the tissue biopsy site, no matter which tissue biopsy site, except for this one in the brain, which we have very few tissue samples, we see that no matter which tissue site you looked at, in the metastatic disease tissue, you can see that these structural rearrangements existed. And you can see that we saw a lot of class 2 alterations. I also do want to mention that one of my mentors back in the day has indicated that MAP kinase is not a target in prostate cancer. So to some degree, I'm almost rebutting my own mentor's work, which is kind of interesting in itself and magical. With that said, I also want to mention that, there's some studies previous that determined that BRAF alterations from a famous New England Journal of Medicine paper by Brandon and Hall indicate that BRAF alterations are enriched in African-American patients. This seems to hold true. Mostly in these patients, we still see structural rearrangement and class 2 alterations. Finally, in Asians like myself, we see that there's also enriched and even more these structural rearrangements and, for example, class 2 alterations. So altogether, I'm just going to rewind a little bit. We took one patient in our clinic and we started exploring these databases, and we're starting to capture the BRAF alterations, either class 2 or fusions, are not unique events that are only occurring in some of our patients. So that said, one of the former trainees in our lab, now a medical student at UPenn, he's helped us characterize BRAF alterations across the GENIE database, which includes far more samples. This is across many, many tumors. You can see what I want to highlight here is that you see, again, blue structural rearrangements, red class 2 alterations. This is a very unique spectrum as compared to all the other cancer types that harbor BRAF mutations. And if you look on the right side, we're essentially putting BRAF on the map, indicating that, yes, BRAF mutations does represent a set of prostate cancer, but this is indeed a small subset. If anything, if those of you think about, for example, ASCO, this almost is kind of like an ALK fusion in the sense that we're targeting a small subset of patients with a specific kinase event that potentially may be druggable in the clinic. Here's some conclusions. The one thing I do want to highlight is that we're continuing to investigate this with some of the laboratory members. We have much more refined, polished data given to us by Tempus, about 500 patients. We will share some of these studies, hopefully in the next iteration. We were taking a look at what type of BRAF alteration, should we reclassify BRAF into different forms, alterations, and then most importantly, we have survival data related to this. We want to bring it to what type of BRAF alterations actually impact survival. We're working with Alex from the City of Hope to really develop this clinical trial at this moment. What else can we find? I want to highlight one case here. David Moline in our lab has been studying a case with Robert Crassi in this large small lung cancer patients. They found is a very unique fusion that is one of a kind. Essentially, we looked at the rates in C-bioportal. Essentially, this represents only about 0.06% of patients have a KRAS fusion event. We don't know what is going on in this particular patient. This patient actually did survive. This tells us there's very unique events in some of our patients that we may be able to think about modeling down the road. And I also want to point out a very unique case study. What maybe some of us are more interested in studying pathology. Here's a case in a lineage patient that exhibit lineage plasticity, as we call in the prostate cancer field. Started with an adenocarcinoma, eventually progressed to had a metastasis to the brain, which is in itself a rare event. Eventually, the brain metastasis infiltrated the actual brain itself. We're able to capture all 3 samples, use and test, get all the transcriptomic data and exome data from Caris testing. You can see here, and this patient obviously did not have a good outcome. You can see the brain lesion here. Interesting enough, this is an event that we're able to characterize using a multi-omic approach. I want to give the Justin Drake lab credit for really characterizing the proteomics part. That's the only part that they needed to do on top of this. That said, we can see that this patient really exhibited a full transition. If you look at the H&E tissue, you see a sort of adenocarcinoma features, and this persisted in the dura once it metastasized, but once it infiltrated the brain, now we're looking at small cell features. AR is completely lost in the brain, and also you can see that mutant TP53 activity by staining is fully enriched in this particular sample, in the brain sample. And you see staining of neuroendocrine markers by pathology. So this, to me, after studying neuroendocrine prostate cancer, is indeed a very unique case where we can capture all 3 phases of a patient. So a lot of my colleagues are asking, can we look at this specific patient, look at the transcriptome, look at, for example, because this is essentially a real progression model that we can capture in our patients. All right, so hopefully I made the case that we can make some discoveries based on our patients. And I only represent a small slew of them. And I don't have time to go through all of them because I really think there's a lot of exciting things that we can learn from our patients. I want to get back to one big question that I think is super important. What do we do with 1 million data points? Because at the end of the day, how do we start thinking about big data points? And to me, this is indeed somewhat magical in the sense that we can start thinking about how to really utilize this data to think about patient outcomes. And we give a few stories and about modeling and how we can think about how to differentiate patients based on gene networks. Now, this might get a little bit complicated. So with that said, if you have questions, please stop me at any time. With that said, I've been really thinking about how genes interact with one another since I was a postdoc. For example, if we look at our conventional approaches, how one gene behaves with respect to one another, we typically look at all the tumor samples, conduct the Spearman-Pearson correlation, and say, okay, they're co-expressed. They have a linear relationship across samples. Well, if we take the next level and use our MSI computational machinery to think about how does a gene interact with every single other gene that we're detecting, and then across patients. So you can see that we're looking at AR and MYC here in normal prostate tissue, AR and MYC in metastatic tissue. So to this degree, all these data points represents another gene. So we're almost mirroring the interactions throughout every single gene to say, how is the gene behaving with respect to every single other gene that we can capture across all the patients? And then we utilize that to model a trajectory to say, are these genes behaving similarly? Are they behaving differently? In this case, we can see a clear difference. Let's just remember, AR and MYC always express somewhat in prostatic tissue, but in the disease setting, they started demonstrating a more linear relationship using this technology, this algorithm that we call ALAN. I just want to mention, for example, we do a pairwise correlation, we only consider 2 genes. Yes, we can somewhat capture this relationship. There's significance. However, you can see that ALAN, because it considers far more entities and sort of interactions, you see a much stronger, and the relationship being the strongest in metastatic tissue. I do want to mention that this is a finding that is not exactly novel. Some other people have shown this based on a lot more time and research. And this is done in many levels. I'm just depicting this model here as a proof of principle. That said, I want to share a few hypotheses of mine going forward, really saying, I think we have to start understanding this. We have to start thinking about this in our modeling, in the sense that gene behavior is not static. As soon as we move to a different tissue, blood, prostate, breast, every time we move to a different disease setting or early disease, for example, prostate versus metastatic disease, these gene networks can change. We have to find a way to account for them. So in order to account for them, then we can start thinking about how diseases progress. And the second one is a little bit more explorative. I've been testing this hypothesis in the lab. I'll share some data to hopefully reflect upon this. Really, we would think that tumor state is a summation of all the gene networks within a set of tumors. In order to capture this state, we can start thinking about, okay, if we can capture all the states that reflect the tumor state, maybe we can demarcate tumors that are from, for example, different patients, responding to certain therapies or not responding to certain therapies, whether it be primary samples, metastatic disease. That's the hypothesis I would go for with testing. How do we do this? For example, this is data that I never presented. This is always inside the lab that we utilize as sort of foundation to think about research. So you guys are the first to see this in public. That said, we think that there's a lot of ways to transform the data to allow us to capture gene networks that are close to another. Remember, Alan's approach that thinks about gene networks allows us to condense gene networks to consider gene networks with relation to, for example, patient outcomes. So that said, for example, this is general UMAP, is a dimensional reduction approach that utilizes the whole transcriptome. It assesses how similar two samples are. Every dot here is a sample. It says, oh, here's a cluster of samples. These are all CRPC samples. They cluster one another. Yes, they're all CRPCs. There's a few outlier clusters here. You can kind of say, oh, okay, there's some different clusters. They may be different. This could be because, quite frankly, the sequencing was really bad. So we just had poor sequencing. This could be also that they're pathologically interesting. They represent a different disease stage. So how do we understand that? What we're showing here is that Allen is a way that can utilize transform. Allen can change the whole matrix to consider every single interaction. That said, we can see now much finer clustering of the outliers. So now we can see, instead of a small cluster to the side, we see a much bigger difference when we consider these sort of outliers. What do the outliers mean? Sometimes indeed, they mean samples of poor quality. But in this sense, we can also capture a suite of tumor samples that have reduced androgen receptor activity, reduced androgen receptor V7. Mainly, again, I'm finding it hard to see on my screen. With that said, we've actually captured almost all but one of the cases which exhibited small cell features or neuroendocrine features. Obviously, these are the most aggressive form of the tumors. But by doing a simple transformation and then a UMAP and considering outliers, we can already capture some features that are based on the transcriptome data alone that relate to the pathology annotation by the clinicians. Here's another remarkable finding that I personally think is magical. And I also want to preface this why I think this is really interesting. Decipher tests, as I mentioned, capture from primary prostate disease a 30% chance at risk of developing metastasis. We can take a look at, again, primary disease. This is all TCGA data from primary prostate tumors. And before transformation of the data, we can kind of see, this is great. We can capture 88% of the patients that never develop metastatic disease, but that is extremely biased because most patients do not. About 10% of patients or less will develop metastatic disease. So essentially, if we use a linear SVM model to predict outcomes, this is, quite frankly speaking, completely useless. With that said, after data transformation, Katie has helped us do this. We can now capture 66% of the cases that eventually progressed. That, to me, is better than what Decipher can do by 2 times. I'm not trying to give us any credit here, but this is definitely an avenue that I would like to explore further with our team. The lesson taken here is that we can use data transformation approaches to clean up the data, the gene clusters, to help us really build machine learning models. Now, how can we do this in real time to think about patients from a clinical trial? Now, we collaborate with Eugene Shanara from Hopkins University, and he also works with the man. I seem like all my connections with the man. I apologize for that. With that said, here's a very interesting clinical trial. They had a very small subset of patients. Essentially, a few patients have been given enoblastutimab. For many of you have heard of B7H3, a drug that targets B7H3. And with that said, some of the patients respond. With that said, there's about 66% of response rate. With that said, you'll see there's about 13 responders and 10 non-responders. This is a very small data set. Can we capture any differences amongst these patients? You see that based on the clinical features, there's no significant differences, mostly Caucasian patients. There's some that smoked, some did not. I'm not sure that allows us to determine who has better response or not, but with that said, none of these values were significant. Now we can take a look at our conventional approaches. This is differential gene expression, GSA UMAP. I'm sure many of you who have operated on sort of informatic data have all done this. And let me just follow up a trajectory a little bit. What would follow downstream of this? Let's say if we consider differential gene expression with responders versus non-responders, we might say, well, they're differential expressed, not significant. Maybe I'll follow up on one of these genes that I have no idea what it does. That might be a research project that goes on for about 5 years, and somebody will be very disappointed. In that regard, we can look at GSA. Again, nothing significant. We have some cutoffs, so nothing quite reaches the FDR at 0.0005. I said, again, a poor postdoc grad student maybe, hey, I'm going to investigate the pathway that's most significant, maybe waste a lot of time. I'm sorry, I'm being a little bit crass here. With that said, we might be spending a lot of time going down avenues where we simply may not have a real answer. And again, we look at a UMAP transformation, all the patients are clustered, well, no, there's no demarcation. However, when we take a look at gene system and understand the concept that the gene are not the same in the responders and non-responders, and this perhaps heat map on the far left is the best indication of this. You kind of see that if I just take the gene profiles after Allen transformation of the AR, the responders for AR, B7H3, and FOXA1, HOXA13, they actually cluster independently from one another. That to me just says that AR is not behaving the same in the responders versus non-responder. Something that differential gene expression does not capture because we're simply looking at if a situation is expressing more or less of a gene. With that said, we're also finding something very interesting. In the responders, we're seeing that there's a much tighter cluster with B7H3 with FOXA1 and HOXB13. This B7H3 stands alone. This is unsupervised clustering here. B7H3 stands alone in the non-responsive patient. It is not linked to AR, FOXA1, or HOXB13, regulators of AR signaling. So this is a different way to depict it, whether with respect to the non-responders or responders. It's a little bit hard to see here, but what I like to argue is that in the red, you can see that there's more overlap with the sort of behavior. This is a model-to-model of behavior. There's more overlap with B7H3 as compared to HOXB13 and FOXA1. Now let's just take a simple Venn diagram. In the responders, we see that there's about 285 entities, features that are overlapping in the responders between the FOXA1 network and the B7H3 network. And you can kind of see in the non-responders, we see almost no overlap, only 3 entities are overlap. These are gene behaviors that we're calculating. So altogether, I think this demonstrates a couple of things. Yes, we can take a very small subset of patients, understand the differences. We can look at the same gene, how it behaves differently across different systems. And importantly, these do behave differently across patients, especially when they're cancerous. An instance which we definitely know that dysregulation of gene behavior essentially is what is driving the disease to begin with. All right. Hopefully I convinced you that there's some modeling that we can utilize to really demarcate patients that are just simply different. Patients that have developed metastatic disease, patients that do not, patients that maybe have aggressive pathological features, and patients that don't. And also lastly, we utilize it to think about patients that respond to certain therapies and patients that do not. With that said, let's think about a way to look at a big consortium of data to model patient outcomes. So with that said, Hannah Bergam has worked with me since I started my lab. So and Dr. Ryan gave us a very interesting case. Essentially, we wanted to study a suite of genes that are in prostate cancer. We see more ligand associated rather as compared to receptor, which Dr. Demps in the audience is an ancestor broadly known as the best androgen receptor researchers in the country. That said, we decided to study the opposite of that with respect to this. For example, androgen production, uptake, conversion. Let me walk us through this process a little bit. So prostate cancer is a disease that's dependent on male hormones. These hormones are circulated. So I'm not a biochemist, but with that said, you can kind of see that the process is fairly complicated. It involves a lot of enzymatic processes. You see that there's some uptake process, production, conversion. The end goal of these enzymes is to really produce a high affinity ligand that binds the androgen receptor, drives transcription and disease progression. So with that said, if you do a string DB mining, so this essentially just takes all literature and it says, oh, what are some connections between these 20 somewhat genes? The answer is they're all connected. What I don't like about string DB is it is heavily biased based on the literature that is inexistent. If somebody has studied that more, that means there's more literature there. HSD3B1, for example, has been studied, especially in the prostate cancer space, is one of the APOC genes that is driving, for example, resistance to androgen therapies. One of the genes, CYP11A1, is actually the target of one of abiraterone, AR pathway inhibitor that's commonly given to patients. This also brought into the world brought into the clinical world based on clinical trials by Dr. Ryan himself. That said, we noticed quickly using a database that some genes are very specific. They're not ubiquitously expressed. You can see that, for example, in the adrenal gland, liver, prostate, testes, these are male hormone related sort of organs. So you can see that there's very selective expression of some of these APOC genes. This told us that, okay, if we're going to develop a biomarker, maybe we should think about genes that are not ubiquitously expressed across patients. That said, one thing we've applied Allen again, and I mentioned that Allen allows us to consider situations that allows us to differentiate gene interactions as dependent on stage. We look at, again, benign prostate tissue, primary prostate tumors, metastatic disease. These are the same suite of 20 genes here. In red, I have labeled 6 genes. So you can see that there's a strong conversion of 6 genes, but mainly in the metastatic disease stage. And it actually correlates with the disease progression. Now, we utilize some of the databases that I have talked about in the earlier part of this talk. We have a collaboration with Caris who gave us access to, for example, 4,500 prostate biopsies and metastatic biopsies, counting about 2,600. So with that said, they did not perform Allen. They used a separate approach. They just did gene by gene correlation. So you can see on the top, you can see here's the 6 APOC genes right here. In red indicates that they're highly correlated with one another. They're not correlated with the androgen receptor, surprisingly. With that said, we also looked at, again, the suite of genes and the metastatic disease. The correlation was more robust. The anti-correlation AR was also more robust. Instead, we found these to be correlated with estrogen receptor, progesterone receptor, a finding that I never would have expected in prostate cancer. That said, these tumors, when they're APOC high, they also have substantially less AR amplifications. This is probably expected. They have less AR-V7 expression, again, pioneered by some of Scott's studies and Emmanuel studies. This is a resistant marker in prostate cancer. So they have less resistant markers, they have less AR amplification, they're less associated AR. To this degree, we might think they might be more indolent tumors. Just a hint there. With that said, we also confirmed this using some standard approaches, just comparing, for example, Mann-Whitney U test. We can see that, for example, all 6 genes in the APOC, which is in red, high, is upregulated as compared to the other group. More importantly, we see stronger elevated expression ESR1, ESR2, and PGR, which we see on the right side. Here's just a confirmation using our machine learning approaches. These APOC genes in metastatic disease are strongly correlated with another see the overlap. Interestingly enough, anti-correlated with androgen receptor, but more associated with ESR1, ESR2, PGR. Now, I mentioned that there's a difference in survival because we expected that they don't have the resistant marks. This seemed to be absolutely the case in the CARES patients. For example, the patients that are APOC high survive 2 to 3 times longer than those that have AR alterations. And if we look at the neuroendocrine prostate cancer patients, these of course perform the worst. Didn't matter if the biopsy was taken in the prostate or metastasis. They all indicated that patients with APOC 6 survive 2 to 3 times longer. I do want to indicate that for those of you who follow clinical trial data in the prostate cancer world, a lot of times we're excited by a 4-month difference. This is almost 2 times much as sort of longer in terms of survival. This to me is remarkable. With that said, you might challenge me and say, well, Justin, these are heterogeneous patients. Can we see this in a phase 3 clinical trial? So with that said, one of our former trainees, Shale, she partnered with her current PI to look at Charted 3, which is a phase 3 clinical trial of patients that have been treated with androgen therapies. Again, they utilize a distinct approach, unsupervised clustering of the transcriptome. Again, found a group that is AR low, APOC high, as indicated in this far right. In this clinical trial, which we see more synchronous patients, the patients are more aligned, we again see that APOC high as compared to AR high has significantly better survival. You can see the difference is quite remarkable again. We're seeing almost twice as long in terms of survival, whether we look at overall survival or progression-free survival. That said, can we take this into blood-based biopsy? I have a long-standing friend that we trained together. We still talk to each other, even though our ideas are sometimes sparse. But with that said, Sullivan has done a really nice sort of epigenomic profiling approach based on cell-free DNA. In a sense, he can measure histone acetylation at the promoters using cell-free DNA, only using 1 ml of blood. That said, we're very excited about it. I asked him, hey, could you please look at the activity of APOC genes in your blood samples as compared to AR expression? So in all samples, we already see a negative correlation. And for those of you who follow the cell-free DNA space, the patients that have higher tumor volume tend to have more metastatic disease and therefore should be the ones that have the AR structural rearrangements and applications. So we surmise that given that the patients with APOC are surviving better, they should be in the lower cell-free DNA fraction, which was indeed the case. If we look at the low cell-free DNA fractions, we can see that the correlation, negative correlation with AR APOC activity is far more robust than in all samples. And if anything, the high cell-free DNA samples, we see that there's almost no correlation at all. We also started investigating why do these APOC patients survive better? This to me is again remarkable because all my teachings have told me that, for example, TP53, RB1, PTEN, these are all factors that drive the difference in outcomes. In the case of APOC, this was not the case. We hardly saw any differences in, for example, statistically significant differences in these alterations of these genes. And also on top of the one thing that we did confirm is that AR alteration is definitely different, along with the several genes that honestly, I have no idea what they're doing. That said, we did see a significant proportion of transcriptomic differences, as is shown by this volcano plot. There's so many differences. There's about 6,000 genes are differentially expressed in these 2 instances. So to me, these differences in APOC 6 versus AR high are mostly driven by transcriptional alterations. Now, I'm just to show this as a final slide. We have deemed that there's a lot of inflammatory pathways that are present and more active in these APOC 6 tumors. We're saying it's hard to see here, but if you look at the absolute fraction via CyberSort, you can clearly see that every one of these numbers mostly is higher in the APOC 6 patients, meaning they perhaps are more susceptible to innate immunity and or adaptive immunity. We did find that, for example, T-cell CD4 naive is about 500 times more in the APOC 6 patients. So, all right, and I talked a lot about how we can engineer transcriptomic data, thinking about with clinical outcomes. Well, I thought the APOC 6 serves a really, cool example of how we can take a suite of genes using algorithm to think about progression, think about genes that only come together in late-stage disease to model outcomes. That says, with that said, I think, this can lead us to many things, many avenues. For example, we can build biomarkers in phase 3 clinical trials, think prospectively. We can also think about other blood-based biopsy options that actually consider activity of these genes using, for example, cell-free chip assays. Finally, we want to think about primary PCs. I assure you that the next set of work that hopefully I'll present at some time is thinking about modeling from the earliest stage of disease and thinking about which of the patients with these sort of biomarkers allow them to progress into metastatic disease as compared to, say, indolent for a decade. That said, I want to make acknowledgement. I cannot do any work without my mentors that are all over the place in this institute and also across other institutes. A great team and invested researchers that really care about the mission and think about patients. So altogether, I will take any questions and hopefully you appreciate some of the magic that we're doing in this particular institute.
Thank you. I've got microphones for anyone with a question.
That was great, Justin. I'm going to ask a question about your boring slide. So that blueprint, that framework, how much of that remains aspirational and how much of that is operational? And how can those in the room help to, break down barriers that might still exist?
Scott, that's a wonderful question. I think we're learning to work with IRB better. We're learning to work with OGC better. I think individuals such as Andrew Nelson, Dr. Internal Records, which we had that discussion during the FCC retreat, have indicated that they will be willing to support us, and they have. So long story short, we have subsequent meetings with OGC to say, hey, listen, this is genomic data. This is not genomic data. Why should you be concerned? What is considered private health records and what is not? I think we started to resolve some of this with the help from OGC, but what I see is the biggest barrier before this was that there's no dialogue between OGC, IRB, and the research and the clinicians. So at the end of the day, it really just required for all of us to be on the same call to really talk about what the clinician needs and how this is not just a research need of, for example, myself. So these are sort of dialogues that were very important to move the process. With that said, I think we're starting to get the Decipher test in. I think one of the instances we're starting to think about Andrew Nelson's test. He said he has about 3,500 samples. We're trying to get the I-SPY 2 data in soon. And with that said, I think the other data set that is looming and very close, which is the Caris data set. We have to do them by order because I just want to mention out here, for example, genetic testing is considered a very different thing because it may be considered identifiable. So any sort of test, for example, Tempus that has genetic data, we have to kind of think through a different protocol with IRB. So that's some of the hurdles that we're facing.
Do you think at the end that the data will flow as drawn in the blueprint, or do you think you'll have to redirect some
Let's go back to this a little bit. I think those are great questions, Scott. And Scott, this is not a boring slide. If anything, it's not even close to boring. It's sometimes
you're the one that called it boring.
It's probably stress-inducing to say the least. But with that said, I would say that here's a part we have to work with more: the Fairview, because Fairview does technically own a lot of the data. So we have to kind of devise partnership with Fairview, which they can help us identify data. Because Fairview, unlike the Minnesota University of Minnesota, is not considered academic. There's different regulations here. So Andrew Nelson has suggested several approaches where they can utilize potentially an informatics team there to help us streamline the process. If anything, I do want to reiterate that the barrier is definitely not from the companies. They're willing to give the data sets. A lot of it is already stored with Steve Johnson.
Justin, that was really fascinating, amazing work. Three really quick questions, comments. Yeah. At the very end in the APUC where you showed no difference between P53, P10, and RB, is that because they're all wild type?
They're all wild type.
Okay.
Yes.
So yeah, and you wouldn't expect the difference then, right? If there was mutations.
In honesty, I was actually expecting a difference. Me and Chuck were doing the genomic, and I was like, it's definitely a difference between that. The APUC higher just don't have as many TP53 alterations RB1, because I thought that was going to be the driving reason why these patients are surviving better. Yeah. So in honesty, when I first got that result, I was like, oh, that's not our hypothesis is wrong.
Yeah.
So
Yeah, no, I think that the inflammation is consistent with so many other things. I think that's really cool. I do like the way that you're creating these sort of transcriptional states in the tumors, because I think one of the things that we've seen as precision medicine has evolved is that people assume that because a mutation is present, it's significant, right?
absolutely.
And they throw drugs at patients that attack that mutation, but it may be totally irrelevant to the disease process. So
Absolutely.
I think this captures sort of what's really happening in the whole complexity of the tissue. Have you had a chance to look at longitudinal samples rather than cross-sectional? And have you thought maybe about incorporating models where you have a little bit more control over the timing of your biopsies? Or your, liquid biopsy or whatever you're doing?
These are really, really excellent questions. And honestly, there are really a lot of prospective research avenues that are currently going after. I'll try to tackle your first part because I think it's extremely relevant. But right now, we're using genomic features to detect, for example, BRAF is mutated to say, oh, there must be BRAF activity. So right now, the current study we're working on with Tempest is they actually gave us the raw data with the transcriptome to see if we can see that the certain alterations are matching to, for example, MAP kinase activity. So, and they have survival data, going forward. They don't have longitudinal sampling there, but we're starting to address some of the questions that I personally think are important that we all are very curious about. Because as you said, sometimes we have passenger mutations that don't drive, or maybe they don't have an effect right now in the disease stage anymore. So I'm trying to answer that question. In terms of sort of serial biopsies, those are still kind of rare to us to some degree. So with that said, I try to track down as many samples as possible to kind of say, but I have to admit, we're still in this sort of, sort of handful setting. For example, there are certain collaborations. We have maybe 10 pre-treated patients, 10 post-treated patients. With the Manny, we're hoping to get some more of these types of patients as I think the clinicians are starting to recognize this as an important thing as well.
Well, just as a pitch, if you're willing to move into the model space rather than the bipedal hairless mammal space, we might be able to have some interesting stuff.
Yeah.
Hey, Justin, great talk. I have a quick question. On one of your last slides, you showed a pretty significant increase in circulating naive CD4 T cells. I'm curious if you've looked at just internal or the intratumoral expression of FOXP3 at all in the patients with really high.
That's a great question. So those are not circulating.
Okay,
so
I misread
they're
it.
they're taken within the tumor biopsy, so we think they're within the tumor. So you may know that prostate tumors are pretty immune cold. So that was a surprising result to me, and I had to talk to my colleague Nick here, but is this weird? But I think once we get some of the tissue samples, that is exactly one of the things that we may go down. I have to admit, some of the prostate cancer researchers, especially clinical trial, they're pretty jaded with sort of immune therapeutics. So some of them are not as interested. I do think this is a research avenue that I might proceed with down the road.
Thank you.
Yep.
Yeah, my question is kind of maybe naive, but that when we think about transcriptomics analysis of a stroma-rich tumor like pancreatic cancer, so we will have to go to the single cell sick, and how much tissue do we need to run those kind of stuff? Because pancreas, it's really difficult to get the big biopsy samples.
Yeah. I think that's a hard question. And I quite honestly speak, I'm not sure if I'm the right person to answer this. I think it depends on what sort of disease states that you're studying. For example, in prostate adenocarcinomas, you have speckles of tumor, right? So I think whereas, for example, in neuroendocrine disease, you have a whole bulk of tumor. So it depends on kind of your question of interest, right? Where are you studying, for example, I work with Julie Ostrander a little bit to study stemness. So in that instance, we want to collect a lot more tissue to think about are there sub-pockets of cells based on single-cell RNA-seq so we can capture those individual cells. So I think, Masato, unfortunately, I'm going to dodge that question and say that it really depends on what you want to study.
And another thing is actually, that I belong to DOS and Department of Surgery. And honestly speaking, that we have to do more prospective sample gathering surgical samples because that the size of a biopsy is not a limit there. Then as a test case, we can do a lot of things with surgical samples. So then later on we can move into the biopsy samples, but I think we have to do more of those kind of stuff.
I definitely agree. Thank you.
Okay, we've got a couple of questions from our virtual audience. Doug Yee, some have considered genomic identifiers the most precise identifiers we have. Thus, sequencing data reveals individual identity. Does this come up in discussions with privacy concerns?
Which part should I answer? How about this? We'll take the first part first. Doug, thank you for always your hard questions. I would say that I think genomic data gives us a layer of prediction. I don't think it's a be all ends all, because if we add in sort of clinical factors, for example, race, ethnicity, it will change the landscape and our ability to predict. There's definitely many other factors. I don't think we can substitute everything with genomics per se. I think it brings us to a certain level of accuracy and prediction. For example, we reached 66%. But my guess is, for example, we think age, Gleason grade, disease state. Don't forget, we did not include that in the model, and this will maybe grossly enhance our ability to predict metastasis. Will they be patient privacy? We are trying to, it's the second question now. We are trying to think about this more. We're really trying to work with OGC and RB to think about at the base pair level, what is capturable and what is not. There's certain determinations that I'm not going to reveal to this certain audience, but I think there's a lot of fine lines that we have to really think about with what is considered identifiable. And then, importantly, Doug, who holds on to that identifiable information? For example, entity like BPIC, they can hold on to identifiable data because they're the honest broker. We cannot as researchers.
Okay, we've got one more question from Yatru Chang. Great talk. I was curious how Allen network size, like 100 or 1,000 genes, was determined. Does the size of the network have effects on the Allen scores?
The answer is yes, it does have an outcome of Allen scores. The network size is determined based on certain statistical thresholds that we deem to be significant. So we set cutoffs because sometimes we realize that gene networks are bigger or smaller. We set very harsh, stringent cutoffs of what we consider within the gene network and outside the gene network. We're realizing that we're not trying to pick the top, for example, the top 100 genes. We're using statistical cutoffs there to model, utilizing that for Allen modeling. So far, we have not found evidence against that, but I agree that there's probably many ways that we can engineer Allen in different ways to kind of think about what are better or worse thresholds. I do think that needs to be coupled with a clinical outcome so that we can have real determination, though.
Okay, are there any other questions in the room? I'd like to say thank you to Justin one more time. Thanks, everybody. See you next year.